Thank you very much to everyone in attendance for joining us. We're really pleased to have three amazing early career researchers with us today. Um, as usual, these will be 15-minute talks, and then we'll have 5 minutes for Q&A. If you want to ask questions after the talks, feel free to just put your question into the Q&A box, or if you'd like to unmute yourself to ask the question, you can raise your hand and we'll call on you and allow you to unmute. But without further ado, I'll start by introducing Sanchirmaa Namjilsuren. She's joining us from the University of Pittsburgh, where she's doing her PhD in the lab of Karen Arndt. She previously got her bachelor's at Southern Miss, and she's going to be telling us a really cool story about PAF1 complex and SPT5. And she just recently, a couple of weeks ago, won the Best Poster Award at the ASBMB transcription meeting for this work, and was given the chance to give a very short notice talk. So she's had a few more weeks to polish it up, and I'm excited to hear what she's got to show us today. Sanchirmaa, take it away.
Okay, first I want to thank the organizers for giving me this opportunity to share my work. And today I'll tell you a story about how the interaction between PAF1 complex and SPT5 governs the requirement for these two essential mRNA 3' end processing factors. And I think this crowd needs no introduction to transcription. So I will just start by briefly introducing the three key elongation factors that we study in the Arndt lab. The first one is SPT5, and it forms a heterodimer complex with SPT4, which is known as DSIF in mammalians. And SPT5 has been widely studied in eukaryotes, and it promotes Pol2 persistivity. It has also been shown to regulate promoter proximal pausing in higher eukaryotes. SPT6 is an essential histone chaperone. It binds nucleosomes and then deposits it back into the chromatin to maintain chromatin structure during transcription. So, and the last but not least, PAF1 complex is recruited to the elongation complex through its interaction with the phosphorylated SPT5. And it promotes co-transcription of histone modifications, specifically H2B monoubiquitination. And these factors, in combination with elongation in Pol2, forms the globally active elongation complex that transcribes nearly all genes in eukaryotes. And I would like to focus on SPT5 because it's the only elongation factor that is conserved across all three domains of life. And what's unique about eukaryotic SPT5 is it contains the C-terminal domain that it consists of multiple repeats of the consensus sequence that can get phosphorylated during transcription. And you might have noticed that this is quite reminiscent of RNA polymerase II Pol2 CTD, which has been widely studied to regulate gene expression through its CTD phosphorylation. But in comparison to the CTD, CTR has been much less studied, but there are varying levels of evidence showing that CTR serves as an interaction hub for multiple elongation factors to interact with the elongation complex. So I wanted to first start off by characterizing what are the genome-wide consequences of mutating the CTR. So I performed ChIP-seq using this SPT5 CTR mutant. First one is lacking the phosphorylation site, so this is the non-phosphorylatable allele. The second one is a phosphomimetic mutant, and delta 5 is a hypomorphic allele that is characterized by its low protein abundance, and the CTR null is only lacking the CTR domain. Here, I'm showing you a spike and normalized occupancy metaplot of RNA polymerase II. And here, as you can see, delta 5 and delta CTR mutants have higher Pol2 occupancy genome-wide. And this is one of the explanations for that is a slowed elongation rate. And because elongation factors depend on Pol2 for its association with chromatin, this varying level of Pol2 can confound their occupancy profiles. So I normalized elongation factor occupancy to Pol2 to show you how much of elongation factors are binding per unit of Pol2. And all SPT5 mutants show reduced Pol2 occupancy. And what's interesting here is SPT5 mutants that is lacking the phosphorylation sites also show reduced SPT6 occupancy per unit of POL2, suggesting that CTR phosphorylation is important for SPT6 retention or recruitment to the POL2. But when we look at the PAF1 complex, it is dependent on SPT5, so it was normalized to SPT5. And the mutants lacking the CTR phosphorylation show greatly reduced PAF1 occupancy per unit of SPT5. On the other hand, the phosphomimetic mutant shows higher PAF1 occupancy per unit of SPT5. Given this profound effect of CTR and its phosphorylation in elongation complex assembly, I wanted to investigate what are the other functions of CTR in general. So using the awesome power of these genetics, I performed a genetic screen to identify negative and positive genetic interactors of the CTR. And to do that, I used this assay developed by the Corman lab at Oxford. And it utilizes a transposase expressed from a gal-inducible promoter and the transposon inserted into an AD2 gene. So when we plate the cells on a media lacking adenine and supplemented with galactose, the transposase expression is induced, and it cuts the transposon out of the AD2 gene and randomly inserts into the genome, and therefore disabling the gene at the site of the insertion. So we get colonies forming when transposase is inserted into non-essential gene, but we don't get any colonies from the transposon inserted into an essential gene. And we can scrape the pool and grow it to saturation to enrich for transposed colonies. And basically, we sequence the region flanking the transposon and therefore map where the transposon went into the genome. But how would we interpret this data in terms of the genetic interaction? So suppose in the wild type background, we get a transposon inserted into gene X, and we get certain number of transposons coming from the gene X. But in the mutant background, the same transposition in the gene X could result in either lethality or a viable strain. In the case of lethality, we would get little to no transposons mapping to that gene X in the mutant background. But in the case of viable strain, we would get either equivalent number of transposons as the wild type, in that case there's no interaction, or even higher number of transposition occurring in gene X. In that case, that means there's a genetic suppression interaction. And this is just a genome browser track showing you where the transposon is inserted. And each gray line indicates an independent transposon, and the essential genes don't get transposition occurring in the gene body. But SPT5 core is essential and the CTR domain is not essential. That's why we get a lot of transposition occurring. And these are the mutants lacking the CTR region. And we can compare the number of transposons in each gene between the wild type and the mutant background and see, okay, what genes have significantly higher or fewer number of transposons in the mutant compared to the wild type. And from here, one gene stood out to us because it had significantly higher number of transposition in the mutant, in all three mutants compared to the wild type. And the genome browser track shows that there is little to no transposition occurring in the wild type, but now in the mutants, suddenly this gene the transposition into this gene can be tolerated. And its binding partner, called REF2, also shows the same trend. There is more transposition occurring in the mutants compared to the wild type. Interestingly, REF2 and SWEET2 are the regulatory subunits of GLIC7 phosphatase, which is known as PP1 phosphatase in mammalians. And these are the members of the cleavage and polyethylation complex. And the GLIC7 or PP1 phosphatase is the phosphatase that dephosphorylates SPT5-CTR. So I wanted to confirm this result by making an independent genetic knockout strains. And I start with the heterozygous mutant diploid and let it go through meiosis. And we use a specialized dissecting microscope to individually separate each spores coming from the diploid and track the growth. And we see that every single colonies that inherited the mutant copy of SWEET2 are lethal, suggesting that SWEET2 gene is essential in yeast. And Ref2 also shows the similar trend, but sometimes the Ref2 mutants can grow like a little tiny pinprick colonies. So this gene is not strictly essential, but it's critical. And I mutated the Ref2 and SWEET2 in the backgrounds of the following SPT5 mutant backgrounds. And I'll just show you a couple of example picture dissection plates here. Here, SWEET2 single mutant is always dead. S2A single mutant grows well. And now combining S2A and SWEET2 mutants are always alive, meaning that this non-phosphorylatable mutant is suppressing the lethality of SWEET2. On the other hand, the phosphomimetic mutant doesn't suppress the lethality. And this is the quantification of the colony sizes from the picture dissection plates. And Ref2 mutant grows a little bit, and SWEET2 is a strictly lethal mutant. But combining these two mutations with SPT5 CTR mutants, we suddenly see a viable double mutants, except S2D SWEET2, because it's a phosphomimetic mutant, and Ref2 can be rescued by S2D because Ref2 is a less severe mutant to begin with. Given that the ChIP-seq result that mutants removing the SPT5 phosphorylation basically greatly reduces PAF1 occupancy, we also tested what happens to if PAF1 mutants also rescue this lethality. And indeed, it does. RTF1 subunit rescues SWEET2 lethality, and PAF1 subunit mutant also rescues the lethality. Even the triple mutant of RTF1, Ref2, and SWEET2 are viable. And this is the quantification of the single and double mutants. All double and single mutants are viable compared to Ref2 and SWEET2 single mutants. And I also complemented RTF1 in the triple mutant using Gal-inducible promoter. And you can see that RTF1 expression in this triple mutant is lethal, meaning that RTF1 is toxic in the background of Ref2 and SWEET2. So exactly what is the function of RTF1 that is necessary for this suppression? And I used the separation of function mutants of RTF1 to test that. And here, mutations ablating the CH2-1 interaction or histone modification domain doesn't suppress the lethality. However, the point mutants in the interaction interface between SPT5 and RTF1 can suppress a little bit. I sometimes get like a pinprick colonies. So I removed the entire +3 domain. Now, the mutant actually rescues SWEET2 lethality. In fact, as PAF1 complex recruitment depends on 3 independent attachment points, and one of them being the interaction between RTF1 and SPT5-CTR. And the other interactions depend on CDC73C domain with SPT6 or CTD linker or the CTD itself. So mutating that CDC73 domain rescues the lethality, but SPT6 mutant doesn't rescue because the domain missing in this allele also is important for SPT6 recruitment itself. And PAF1 complex being a positive elongation factor, it's known to reduce elongation rate. So we used a mutant called 2 RPB1 point mutants that have faster or slow elongation in vitro, but it both has slow elongation in vivo. And just slowing the POL2 speed can rescue the SWEET2 lethality. And I briefly mentioned that Ref2 and SWEET2 are the regulatory subunits of QUIC7. So we wanted to know what happens to QUIC7 in this triple mutant with the RTF1. And I performed a ChIP-seq to look at POL2 occupancy and QUIC7 chromatin occupancy. And here, the mutants lacking RTF1 have great increase in POL2 occupancy consistent with elongation defect. But if we just look at the QUIC7 occupancy, QUIC7 is normally enriched at the 3' end of genes, but with the deletion of Ref2 and SWEET2, now QUIC7 is depleted from the 3' end of genes. And this is just an example genome browser track showing the enrichment of QUIC7 at the 3' end of gene and its absence in the Ref2 and SWEET2 double mutant. And this is showing you that depletion across the genome just from the CPS, QUIC7 is depleted. So our current working model is, in the absence of SWIT2 and DREF2, QUIC7 is dislodged from the CPF, and therefore it causes hyperphosphorylation of spt5. And the spt5 phosphorylation being responsible for binding PAF1 complex, we think that this hyperphosphorylated spt5 holds onto PAF1 beyond the CPS and therefore causes lethality in this double mutant. And non-mutually exclusive model is that slow elongation rate in the absence of PAF1 recruitment is also suppressing this defective mRNA processing machinery. And with that, I want to thank my advisor, Karen Arndt, for all her support and guidance throughout my PhD. And I want to thank my committee members and our joint lab meeting groups for their shared resources, and all these other labs for sharing plasmids, strains with us. And the Rainish lab, who we are having a collaboration on a biochemical assay to identify CTR interacting proteins, which I didn't have time to talk about in this talk. All right, and I'd be happy to take any questions. Thank you.
All right, great job. That was fascinating. I'll start while we wait to see if any other questions come in. It's really curious, all of the mutations you're identifying are complexes that are associated with, positive elongation. I'm wondering from the screen, are there any other essential hits that you'd like to talk about, or that you are willing to talk about, or is it?
Yeah, so there are some interesting hits from the screen that will be included in my manuscript. I also followed up on some splicing factors that have very interesting interaction with the CTR. It's not exactly synthetic lethal or suppressive interaction, but rather the suppression of its SPT minus phenotype, which we are still trying to figure out.
And so now we've got a question in the Q&A from Maria Aristizabal. Very cool story, great job. Based on the model you presented at the end, do you think the defect can be rescued by mutating PAF1 so it can no longer bind the CTD?
Yes, that's the expectation. It is with the CDC73 mutant that we used, that domain in the CDC73 interacts with POL2 CTD. So we're expecting that in that mutant, the CDC73 cannot interact with CTD, and therefore its recruitment to elongation complex is impaired. So it does rescue the lethality.
All right, well, then if there are any other questions, please go ahead and continue to put them in the Q&A, and Sanchirmaa can monitor that.